Okay, so chapter 4 is the one that concludes our first overview of living things, and it's a little bit of a tour of the cell. If we want to find out what exactly cells are and why people even became interested in them, one of the things we could do is look at history and see where all this knowledge ultimately came from. And it takes not a rocket scientist to figure out that very often when humans do things, there is a certain motivation required. And the motivation for cell research really came from various types of illnesses. In particular, in the Middle Ages, the plague was a big problem, and it had spread across Europe, and people could not explain what was behind it. And when people have sort of an emergency situation, that's sometimes when research really picks up. That's when you have a motivation. This is what happened recently when people in the 1980s discovered the AIDS virus. Right? There's this virus, and nobody really knows what it does except that people die from it. So what you do is you try and figure things out. That's the science method. And as a consequence of the research that began on the AIDS virus, we developed an entire new field of research called molecular biology, which then in turn helped with a lot of other things. And so it was with the emergence of cell research. People simply started to look at look at small things in ways that people had never been able to before. Now, there was an interesting part of this. People had started at that time also to look out into space. And so the research that was taking place when people were starting to go out into space also helped develop some of the technologies that were required to look at very small things. In 1665 is when Robert Hooke had a piece of equipment that was useful to look at very small things, and he was able to see something that he likened to the cells in a monastery. That's what he thought he saw. It was an orderly arrangement of very small features in, at that point I think it was a leaf, and he called these cells. Now, cells are very small compartments. I mentioned this before. They're filled with primarily water and a variety of other chemicals, and they are the things that all organisms are built from. They are necessarily small because if you consider the amount of work chemicals have to do and the small size of these chemicals, you don't want to have chemicals travel huge distances because that means that they might be late to where things are needed. And so it is a function of the chemical need of the cell that determines ultimately how small or how large a cell should be. As part of Hooke's discovery, people identified that there were a lot of interesting things at the microscopic level. And so it's a little bit like a bandwagon. Once somebody finds something cool, then a lot of people want that experience. A lot of people have that interest, and a lot of people starting to work on that new technology. And so many scientists began to use these very simple microscopes. They began to look at tissue structure, not only in plants, but also in animals. And of course, you start with anything you can get your hands on. Find an insect, put it under the microscope. Find a piece of skin, put it under the microscope. Anything that works. And so people started to accumulate a lot of evidence. Most of this evidence was discovery type of evidence. It was descriptive. People published drawings. And some of these drawings from way back when, they're still extant. The books are still around, and they're very good. And it turns out that all organisms had cells. This is very interesting and very important. If you find something that is so fundamental to all living organisms, there must be something really important about that. As a consequence, they began realizing that this was a universal thing about living organisms. They all had cells, and so this developed something called cell theory. Cell theory simply says all living things are constructed of cells. That's sort of a first part of it. And then, and this is sort of an interesting one here, all cells are derived from previously existing cells. Now, this is a little bit tricky because when we talk about cells derived from previously existing cells, it becomes a chicken and the egg question. which was first. I mean, how can you have a cell if there wasn't one before? Well, this would obviously say you can't. So at some point, a cell, the first cell, must have come from nothing. Well, that's an issue, right? I mean, right when people started to develop some understanding of what cells were and what kind of things you can do to observe cells, then they ran into this fundamental conundrum. And it took them a while to solve this. It will also take us a little while to solve this. We're going to wait until a later chapter. But in the meantime, we're going to try and figure out what cells are. Because if we know what cells are and how they operate, then we can come back to this issue of cells being derived from previously existing cells and tackle this with a little bit more knowledge. So that's what we're going to do. We're going to look at the differences in cells. We're going to look at the different cell types. It turns out that there are two major cell types: prokaryotic cells and eukaryotic cells. Prokaryotic and eukaryotic are the two major cell types. When it comes to how a cell is built, there are some common structures. And this is, again, very interesting. All cells have a plasma membrane. This is also something that people have called you probably guessed it, cell membrane. It is, after all, the things that's around the cell. So it's the cell membrane or the plasma membrane. All cells have DNA. So that's an interesting feature. And all cells have certain structures called ribosomes. And ribosomes are the things that assist in protein construction. Now, you have to think about this for a minute. This is all cells. This is bacterial cells. This would be yeast cells. That would be your cells. All cells have these features in common. Every single one of them. And that's pretty fundamental then. It tells you something about cells. If all of them have it, that probably means all of them need it. If they don't have one of these parts, then maybe they can't be cells. So just as we were identifying some very fundamental concepts about the molecules of life, now we're starting to realize there are also some very fundamental things about cells. Here are the two cell types juxtaposed. They are very different, as you can see. One of the big differences is size. And if you remember that cells are, of course, three-dimensional, then you realize quickly that one small prokaryotic cell like this one fits into one of these larger cells, not just perhaps 50 times by drawing it all along to cover the surface, but there is also a third dimension to it. So you'll find that prokaryotic cells may be 1,000 times smaller than eukaryotic cells. Well, that's obviously a big difference. And one of the questions we may ask is, well, how did they get to be that big? I mean, where did this transition occur? How did this even happen? You can also clearly see that inside of the eukaryotic cell, that large cell, there are a variety of structures that are missing in the prokaryotic cell. The only thing you can really see in this prokaryotic cell is this stuff in here. That's the nucleoid region, and that basically contains the cell's DNA. So it's a small area, which means it probably doesn't have a huge amount of DNA. In contrast, look at this area here. This area here, that is the nucleus of the eukaryotic cell, and that's where its DNA is. It stands to reason, if that nucleus is so large, it probably has quite a lot of DNA in comparison. On top of that, there are a variety of structures outside of that nucleus, like this, and of course like these. There are all kinds of little compartments, and these compartments are called organelles, and we'll talk about those in a few minutes. If you just wanted to compare these cell types and come up with some very basic conceptual differences, you can say, well, the prokaryotic cell is very small. It does not have a nucleus. It has no organelles. It has a cell wall that helps it keep its shape. And they operate independent of other cells, meaning that they don't form anything like tissues, like what we have in our body. And lastly, these are the oldest kinds of cells. We have fossils of prokaryotic cells that are in rocks that are approximately 3.5 billion years old. Secondly, the eukaryotic cell have these organelles, and it turns out these organelles are themselves surrounded by membranes. So that's a level of complexity that we don't have in the prokaryotic cell. Yes, each prokaryotic cell has one of those cell membranes around it, but that's all the membrane space you have there. In the eukaryotic cell, not only is the entire cell surrounded by this cell membrane, but there are also all kinds of structures on the inside that have membranes. So there's obviously something about membranes that is important to these large cells. Perhaps one might hypothesize that it is in part because of these membranes that a cell can get so large. And we'll talk about that in the next segment. There is a large nucleus, and the nucleus really dominates the cell. It dominates the cell not necessarily just because it's large, but because of the DNA it contains. The nucleus is the driving force of the cell. There are many structures inside of the eukaryotic cells. There is a division of labor among the organelles. They can actually get together and form tissues or other types of features where multiple cells get together and work together as a group, sometimes a very tight-knit group, such as in our organs, but also sometimes outside in cell complexes. And these cells are younger, quite a bit younger. 2.1 billion years ago is when we see the first appearance of eukaryotic cells. So there are a lot of differences about these. Having these differences means that these two cell types are going to be producing different kinds of organisms. One kind of organism that's going to be very small, relatively simple, those would be the prokaryotic organisms, and one that has the potential to become more complex. That would be the eukaryotic cell. And it takes, it's not very difficult to figure out that our own cells must be eukaryotic cells because we are a pretty complex organisms. Now let's take a look at what types of organisms we can produce. Irrespective of what cell type you have, you can produce unicellular cells. Unicellular cells are those that are composed entirely of an individual cell. So a single bacterium would be a unicellular organism, or a single yeast cell, or a single amoeba. Those would be examples of unicellular organisms. Multicellular, of course, is the many-celled organism, and a many-celled organism consists of many cells, and that would be us. Now, as before, one of the things I want to point out to you that these words tell you something. Uni means one, multi means many. So if you're wondering about this in terms of, what does the name mean, this is what it tells you. One is uni and many is multi. Now, the unicellular organisms include certain groups of organisms: bacteria, archaea, and protists. Now, the bacteria and the archaea, those are organisms that are composed of prokaryotic cells. These ones here. The protists are the ones that are eukaryotic. So even amongst relatively simple organisms that are composed only of a single cell type, you find these differences. Protists are eukaryotes, and bacteria and archaea are prokaryotes. And then, of course, you have multicellular organisms, the kingdoms of life that you've probably heard of, plants, fungi, animals, and all multicellular organisms are eukaryotic. All are eukaryotic. Well, that tells you something about prokaryotic cells. It tells you that they can't do this, and therefore they are limited in what they can do. They are limited in how they can cooperate, if at all, and they're limited because they have relatively simple internal structures, which means they can't grow to be particularly large. So those are the types of organisms. Let's take a look at one of these. Here is an idealized prokaryotic cell. Idealized because it's a drawing, and as I'll show you the picture in a little bit, you can't always see things so well in a picture. Now, it has some structures that are used for locomotion and attachment. Those would include the prokaryotic flagella. The prokaryotic flagella are embedded in the cell membrane and the cell wall. Green here, there's a membrane, and then around it is a cell wall. And there is a way to embed these flagella so that they can turn. And when a flagellum like this turns, they can actually turn 360 degrees. They literally spin round and round and round. That's how they work. There are also these smaller structures called pili, and these are bristle-like structures, and in terms of bristle-like, they're not like a hairbrush bristle-like, but they're small, they're fine, and they're sticky. And that means an organism like this might actually stick to the surface of, let's say, your stomach lining, because all of this stuff here is sticky. And these pili and the bristle-like structures, they are the ones that are allowed to do that. So they're called pili. Now, when we go from the outside in, the first thing we have on the outside is a layer that allows these cells to remain moist, and that's the glycocalyx, and that's this orange layer here. This orange layer is made up of some type of carbohydrate substance, and glyco is basically the word for that. Glyco, remember glycogen or glucose, it's like that. So it's a sugary capsule. Calyx means capsule in Greek. So you have a sugar capsule surrounding the cell. Prevents it from drying out. It's an added layer. It sometimes helps with the stickiness. The next thing is the cell wall. The cell wall is what gives it, the prokaryotic cell, its shape. And then on the inside of that is that plasma membrane. So there are 3 layers before you can get to the inside. Now, there isn't much on the inside. On the inside, there's, of course, the cellular juice. That's the cytoplasm. And in addition, inside of that cellular juice, we have 2 structures that are noteworthy and noticeable. One is the nucleoid region. That's what has the genetic information, the DNA in it. And then these little small ribosomes. And ribosomes, as I mentioned before, those are common in all cells, and they help make proteins. So that's what an idealized prokaryotic cell looks like. And now let's take a look at one in an actual image. Well, here is the prokaryotic organism. It has, as you can see, these flagella. So those are the 3 flagella we saw in the previous picture. It has those. But beyond that, you can't really see much at all. Well, sure, okay, you can see that there is this layer here. That's the layer we call the glycocalyx. That's sort of the sugar layer, right? Glycocalyx. So that's there, okay. And then we have this greenish layer, and that's the cell wall. So all we can really see is the cell wall, but we can't really see anything else. And there is a limit to what we can see about these anyway, because they are so small. So we can't see any of the stuff inside very easily. We can identify a few structures, but that's about it. Now, from that relatively simple kind of organism, we go, bam, to this huge and complex animal cell. Now, I'm not even going to go over all these individual labels. I just want to give you the impression that this is vastly more complex. There are some significant structures in there. There are a lot of layers, a lot of bits and pieces, a lot of floating bits. There are many different types of organelles. There is also a flagellum, but it's different. This looks more like a tail. It's much thicker. It's a differently structured type of thing. And so the animal cell is very different in what it looks like. And even if you look at an actual diagram, this is a paramecium. It looks a little bit like somebody's slipper or the bottom of somebody's shoe. It is a unicellular eukaryote, so it is made of all those things that I showed you in that previous diagram. I don't want to identify them all here, but just a couple of them so that you can see. This large one here, that's the nucleus. And then there are a variety of organelles. There are some that have things in them. I'm not going to identify them, but you can just see there are some with content, then there are some that don't have much content. Maybe those are for storage. Then around the edge, it has these little cilia. That's what replaces the locomotory structure in these. It's not one big flagellum, but it's actually many smaller kinds. And then there's all these other little things that are not easy to identify in the middle of it. So clearly, this is much more complex. Now, if you compare that to the plant cell, you can see that the complexity is still there. Plant cells and animals have the same type of complexity. There are really only two things that I want to point out to you about the plant cell. The first one, and that's a clear difference, is here. There is an actual cell wall. It's also labeled here. That cell wall is absent. And of course, we know that plants have sort of harder surface areas than we do. They're not as malleable. And if you think of a tree, well, that's certainly got a lot of hard parts to it, right? So the cell wall is important in building those kind of structures. And then, very important here, is something called a chloroplast. And the chloroplast is an interesting organelle because it is the organelle of life, if you will. It is what allows the plant to attract sunlight, to harness the energy in the sunlight, and then convert it to structures that we can all use. So these little things here, these little green bits, we'll talk about those in chapter 7. They're going to be very important in terms of building a basis upon which life can evolve and operate. So this structure here, the chloroplast, is going to be something that we'll spend a bit more time with. Now, the last category that we're talking about in this discussion on cells is one that isn't really a cell at all. We put it in here because viruses are a very important part of our medical history. They are certainly something that have very recently become a very important part of the news. If you think of the Ebola outbreak in Western Africa, it's stunning how a small structure like this, a virus, can spread and within a couple of months kill thousands of people. And this isn't even a proper cell. Viruses aren't actually organisms. They do have some characteristics of organisms. For example, they do have genetic material. In some of these, that genetic material is just like ours. It's DNA. But in some of them, it's not. In some of them, it's RNA. So there is a fundamental difference in how these things operate compared to how we operate. They are non-cellular, meaning they don't actually have the features of a cell, so they don't have a cell wall or a cell membrane. They have what's called an envelope, a membranous envelope, which doesn't operate like a proper cell membrane. So there's the membranous envelope. In some cases, they just have a protein coat. These are just individual protein molecules that are stacked together, and that's what it has. So that's not a cellular structure. They also are unlike cells because they cannot reproduce the way cells do. Remember, it said cells come only from cells. Well, viruses all by themselves can't do anything at all. They just sit there. They need the cellular environment of an organism to do anything. So you can have this Ebola virus sitting on the table and it won't do anything unless somebody breathes it in. And then it becomes a serious challenge. So they cannot reproduce by themselves. And so rather than considering them cells or organisms, we should probably best consider them as parasitic particles. That's really are. Just like dust is a particle, a virus is a particle, but it is a particle that can have some serious effects on you. And so I don't want to spend a huge amount of time on viruses, but it's just to show you how fundamentally different they are in comparison to the prokaryotic or eukaryotic cells. And that's the end of the first part.